In addition to looking at gene expression, you could also be interested in a marker or a mutation, and you want to know how it is expressed in the patient population you are interested in. In LandExplorer, powered by OmicsSoft team, you can use custom queries to generate a chart like this. Here, you can see how mutant and wild-type TP53 is expressed in colon cancer, and you can use this information to generate survival curves to see how expression of your gene of interest or mutation status influences survival of patient populations with colon cancer, for example. Let's imagine you conducted a genetic analysis on a small number of specimens from patients with colon cancer and found mutations in KRAS. Based on these findings, you want to know how the expression of your gene of interest, ICAM1, is associated with KRAS mutations in colon cancer. To look at how gene expression is associated with mutation status, you can search for your gene of interest under the Genes and Chemicals tab and click on the hyperlink under the Symbols column. This will open up the GeneView page in your browser, which will have additional information about your gene of interest. Here, many users like you focus on the section that has OmicsSoft. As I mentioned earlier, OmicsSoft is a group that's processing all sorts of public data from sources like Blueprint, CCLE, GTEx, and many more. Here we'll select TCGA. This launches LandExplorer, which is a nice integration between IPA and OmicsSoft, where we have data for tens of thousands of samples all processed using the same pipeline. Here, the expression of ICAM1 is displayed across different types of cancers, including our cancer of interest, colon cancer. Since you want to focus on colon cancer, you can use the filters on the left-hand side to filter for your cancer of interest. Under tumor type, check None, select colon cancer, and then click Apply. Once we do this, everything will be removed except for colon cancer. On the right-hand side, you can see what types of tissues the data is coming from. Next, we're going to create a mutation query for KRAS by going to the Customs button and selecting DNA somatic mutation. Here, we're going to search for our gene of interest, name our query, and click OK. Once we do this, you can see that our custom query appears here, and you can use this information to remove the missing by checking on Missing and then clicking Apply. To see how ICAM1 and KRAS mutation status are associated, we're going to go ahead and change our grouping from tumor type to our new KRAS mutation query. This will generate KRAS wild type and KRAS mutant groups. If you want to know what these samples are, you can select them by drawing a square. Once we do this, a table will open up down here that will tell you the TCGA numbers for the samples, will give you information on copy number variation, and will give you expression values as well. Many times, users like you will download this data from LandExplorer to Excel using this button. They will then use this data in downstream applications or use it to make their own custom chart. Now, because KRAS is a marker of poor prognosis for many cancers, you want to learn more about the patient survival for those diagnosed with colon cancer. To see how ICAM1 expression is associated with survival, we're going to go ahead and change our view from gene FPKM to survival data. This view shows a plot of percent survival across time. The y-axis represents the percentage of subjects alive, while the x-axis represents time in days. In addition to this, you want to know what is the survival for colon cancer patients with KRAS mutations. To do this, we're going to go ahead and change our grouping from tumor type to our new KRAS mutation query. Now you can see how KRAS mutations are associated with survival in colon cancer. You can click here to show statistical values. If you want to share this chart as an image, you can export it by clicking on this button right here.